All right, good morning, afternoon, evening, everyone. My name is Martin Hurst, and I'm a professor in the Michael Smith Laboratories at the University of British Columbia. And it's my pleasure to serve as your host for today's seminar, kicking off the 2025 season. Before we begin, I'd like to take a moment to acknowledge the Indigenous people on whose lands we are gathered today, wherever we may be located. This recognition reminds us of our shared responsibility to honor the land and our connections to one another as we work together towards realizing our collective aspirations as a nation. As a reminder, and as you know, today's seminar is virtual, and I would ask that you pose your questions in the Q&A box. You'll see that at the bottom of your screen. There's also a chat function, but for questions, please do use the Q&A box. Now it is my distinct pleasure to introduce our first speaker, Dr. Mathieu Schulz. Dr. Schulz completed their PhD in 2022 at the PCL in France under the mentorship of Dr. Deborah Borges in the Institute Curie. Their doctoral research explored the role of DNA methylation in early fate decision during mouse embryogenesis. Through this work, Dr. Schulz developed expertise in genome editing and genomic analysis, generating DNMT triple knockout mouse embryonic stem cells and employing advanced techniques like cut and run whole genome bisulfite sequencing and ATAC-seq to study differentiation and its impact on the genomic landscape. In 2023, Dr. Schulz joined Dr. Kimmins' lab as a postdoctoral researcher, transitioning to human models while continuing their focus on epigenetics. Their current research investigates the human sperm methylome, specifically how it is established during male germ cell development and its role in paternal epigenetic inheritance and offspring development. Today, Dr. Schulz will share their latest findings on how body mass index influences the sperm methylome and its implications for early human development. Dr. Schulz.
Thank you very much for this introduction. I will start sharing my screen. If people can see it right now, let me know.
Yeah, looks good.
Yeah?
Yeah.
Perfect. So thank you all for attending this seminar. I would like, first of all, to thank the organizer from the CERC network and Active Motif team for allowing me to present my work today. So I will speak about one of my projects that I've been starting in Dr. Kimmins' lab at the Recherche in the University of Montreal, which aims at understanding how a father's BMI can affect offspring development through the sperm DNA methylome. So, as you may already know, obesity has been qualified as a global epidemic by the World Health Organization, and close to 1 billion individuals in the world now live with obesity, and it's estimated that 2.5 billion are considered overweight worldwide. The rates of obesity in adults have basically doubled since the 1990s. And for our information, these rates of obesity have more than quadrupled in adolescents at the same time. With this, human association studies have found that paternal BMI, an increase in paternal BMI at conception, is associated with an increased risk of fathering children that will develop neurological disorders, such as autism or ADHD. Overall, the rate at which these public health issues are emerging are too rapid to be solely attributed to genetic variation, natural selection, but rather originates from a complex interplay of genetic and environment lifestyle factors. So how can we possibly bridge that gap? Well, in recent years, epigenetic modifications, being dynamic and reversible markers of genome regulation that are known to be shaped by the environment, have emerged as a potential platform for the transmission of non-genetic information from parent to their offspring. For example, DNA methylation, which consists in the deposition of a methyl group on the cytosines by the DNA methyltransferase, is a well-known epigenetic mark that is particularly sensitive to regulation of metabolic pathways such as the folate cycle or the one-carbon cycle, and is also a critical regulator of spermatogenesis in human and in mice models. For example, in humans, a previous study from our lab has detected impaired sperm DNA methylation signature in population of South African men that were exposed to increasing dose of toxicants such as DDE, which is a mosquito repellent. And in those population, these men have a higher rates of birth defects. So despite the advancement in understanding the sperm methylome, previous research mostly focused on gene promoter and imprinted regions for good reason, but tend to overlook the intergenic, more dynamic and underexplored regions of the genome, and leaving an incomplete picture of the relationships between BMI and the sperm epigenome. Additionally, the link between the sperm methylation signatures and early human embryonic development is often neglected. So in this study, we used a panel array that was developed in the last few years by our collaborators called METHI-C capture sequencing. It's a tailored panel of capture of sperm region with intermediate levels of DNA methylation or intergenic region that are thought to be more sensitive to environmental changes and potentially act as biosensors. Currently, our dataset includes 52 sperm samples from men that showed normal semen parameter according to the World Health Organization standard. And the dataset is divided between 20 normal BMI individuals, 20 overweight, and 12 obese individuals. So we wanted to understand what would be the impact of an increasing BMI on individual CPGs. To do that, we applied a general linear model on BMI adjusted for covariates like age, and we try to detect differentially methylated cytosines, or DMCs. So those would be cytosines that would show a significant difference in methylation in the obese and overweight individual versus the normal individual. And then we define differentially methylated regions, or DMRs, that for the same significancy threshold accumulate 3 or more DMCs in a 500 base pair window. So genome-wide, we detected over 38,000 differentially methylated cytosines in the sperm of men with an increased BMI. The vast majority of them tended for hypermethylation, with 34,000 of them being hypermethylated with increased BMI. Among the genes that accumulate the most significant DMCs, we found some interesting targets that I would like to quickly mention. We have the myelin transcription factor like 1. We also have ADAM-TSL3, a gene for which variants have been identified in disease such as diabetes and schizophrenia. And on the other side of the medal, for the lost DMCs, we can mention catenin alpha, which is a gene that has a strong brain testis expression and is involved in early embryonic development. Overall, these DMCs tend to be significantly enriched at intergenic regions like we suppose they would be, but we still find a good proportion of these DMCs to be located at upstream TSS, promoters, at exons, and at UDR. So if we check at those genic DMCs, and if we try to see if we can detect some differentially methylated regions that would overlap at those genic regions, we found over 3,800 sperm BMI differentially methylated regions that would be located at above 2,030 genes. When we run a gene ontology analysis, we see an overrepresentation of neural pathway, especially relating to synaptic transmission and organization. But disease ontologies also shows an enrichment for pathways that would associate with the development of autism spectrum disorder and autistic disorders. So all of these findings are consistent with previous research that shows an increasing risk for sperm to influence neural development in the next generation. But how could these DMCs and DMRs influence the next generation? How can they be transmitted? So to develop that idea, I would like to quickly talk about transposable elements. So as you may already know, transposable elements are repetitive sequences found across the genome that have the ability to jump around and multiply themselves. And their activity is actually tightly controlled by DNA methylation, especially in the male germline. But what is interesting with transposable element is that their expression is sometimes allowed and relate to very specific timings of development. So for example, at early human embryonic development, at the time of zygotic genome activation, we can see the activation of line elements and some ERV element families. What is also interesting is that some of these families of transposable elements also resist the wave of epigenetic reprogramming that happens in early embryonic development, so potentially allowing for the perpetuation of epimutations from one generation to the other. For example, from the fertilization to the blastocyst formation, the genome-wide level of DNA methylation usually falls below 20%. But we can see that some families of transposable elements, here, for example, line 1 element or the subfamilies line 1PA, like 1ME, in the human embryo will keep an elevated level of DNA methylation up to 50%. And indeed, when we look at the DMCs in our dataset, we see a strong enrichment for the accumulation of differentially methylated cytosines at ERV element and line 1 element. And we can retrieve again those subfamilies that I was presenting you quickly. So we have the line 1 PA13 family, the line 1 PA6 that are active in the early human embryo. The line 1 PA elements have also been implicating in long-range gene regulation. We also find some SVA, some ALU elements that are known to be critical for zygotic genome activation. That is because transposable elements are bearing in their sequence the binding motif of many transcription factors that are thought to be important to regulate gene activity at specific time points of development. So we wanted to know if those sperm BMI DMRs that we found at transposable elements would be associated with regions that are becoming active in the early human embryo. So we used a public data set that was listing all active enhancers at human ZGA. And we found 197 DMRs in the sperm that are responsive to BMI that seems to overlap with those ZGA enhancer and regulate around 151 genes. Interestingly, among those 197 BMI DMR, 177 of them are also overlapping with the transposable element, again showing up the alusine, line, and ERV families. These BMI DMR at ZGA enhancers that bear a transposable element show binding motif of transcription factor that are critical for embryonic development. So I would like to highlight the OTX factor and the PU3F3 factor that are critical regulator of brain development and embryonic development. What is also interesting is that those specific subset of ZGA enhancer that we have found overlapping with the sperm BMI DMR seems to retain an active chromatin landscape from the timing of ZGA to the timing of the blastocyst. So you can see here that they retain open chromatin and active chromatin signal from the 8-cell stage and ZGA to the blastocyst stage. So we wondered whether the sperm BMI DMR could also impact important genes that would be critical for lineage decision down the line at the blastocyst stage. So once again, we used public RNA sequencing of human embryos performed in the embryonic ICM lineage and the trophectoderm extra-embryonic lineage. And we found 477 sperm BMI DMR that overlap with an active gene promoters. So that sums up to around 186 genes. We observed also that the vast majority of them tend to be more expressed in the trophectoderm lineage. And indeed, the binding motif of the transcription factor that would bind to this promoter highlighted the role of TFAP transcription factor or ETV factor that are critical regulators of the trophectoderm. This highlights the hypothesis that we could have an altered trophectoderm development via inherited sperm BMI DMR. And this is in line with observation that we have higher placental defect in pregnancies from father that have an elevated BMI. So to give you a couple of take-home messages that sums up this presentation, we found that increased weight in men affect the sperm epigenome at over 38,000 CpG sites. And we found most of them at distal intergenic regions. I didn't mention this in the presentation, but I would like to highlight that we also found some differentially methylated cytosines or regions that are not only affected by BMI, but are also sensitive to different cues, such as age or increasing age, meaning that we have a set of sensitive DMCs and DMR that are sensitive to multiple external cues. So that would be an interesting thing to pursue in our study. We found that the transposable element, and especially line, ERV, and ALU elements, accumulate BMI DMCs at the sperm and associate with region that tends to become active regulatory region at the timing of ZGA. Since we observed that these elements and some gene promoter bearing some of these DMR tend to be more active in the trophectoderm, we would like to suggest the idea of a sperm to placenta to brain development axis, where inherited BMI DMRs from the sperm would affect the placental development, and the placental being a very critical regulator, a neuroendocrine tissue regulating the neural development, could affect offspring neural development as well. It is well known that the paternal genome, epigenome, is a critical regulator of extra embryonic tissue in supporting the development of the offspring. And so our observation fit into that theoretical frame. I would like to conclude this and thanking you all for your attention. And I'm ready now to answer some questions.
Wonderful. Thank you. Thank you, Dr. Schulz, for that fantastic talk. Just as a reminder for everybody that the Q&A is open, so you can ask questions via the Q&A box, and I will be happy to pose them to our speaker today. Maybe just to get things kicked off, I'm going to ask us a provocative conceptual question, which is, evolutionarily speaking, why do you think this mechanism is there? In other words, could it be that this mechanism of altered DNA methylation is actually a biological protective mechanism rather than something that's propagating disease? And if not, then why do you think that this would be something that was retained in the primate lineage?
So that's a great question. I love all things related to evolution, and sometimes it is hard to make bridges, but I think there's some interesting observations. First of all, in the sperm, we do have regions that are very, very, very tightly regulated for their DNA methylation level. Obviously, the first example is imprinting and gene promoter that have either a very strong hypermethylation profile or a very low hypomethylation profile. And those regions tend to not be affected as much by environmental influences. So these regions are most probably more resistant to those variations. And that was the whole rationale between trying to look at more intergenic regions that have more heterogeneous level of methylation and that could be more sensitive to those regions. So I think the very critical regions are very well protected from development and from modifications of the methylation. And then you can also think that in early mammalian development, you have this wave of DNA methylation erasure that is drastic and genome-wide that could also act as a fail-safe to avoid as much as possible those passing through generation. And that's where the transposable elements are interesting, is that transposable elements tend to resist these DNA methylation wave. And many studies have found that they could act as potential sources of innovation and new genetic control and promote this kind of genetic diversity that could, like, later on evolution play a bigger role. So if we take the road of the whole placenta, you have the whole placenta is basically due to the activity of syncidin, which is a transposable element like protein. But with that source of innovation comes the risk of also possibly transmitting epimutation or genetic mutation that could then associate with a higher risk in the offspring.
Yeah, thank you. I think it's a very kind of intriguing
Mm,
conceptual question to think about protective versus deficit, and why would that be encoded in such a way? There's
that's
some
right.
great questions. There's some great questions coming into the Q&A, so I'm going to have time to ask a couple of them, and then we will, or Mathieu will be able to answer the rest offline. But we do have another fantastic speaker today. So the first question is awesome talk from Genevieve. She asks, it seems that you have differences in subtelomeric regions. Uh, she apologizes. Sorry, it was too fast. Can you speak a little bit to some differences that you might have seen in the subtelomeric regions?
Yeah, so absolutely. So in the Manhattan plot, we see a lot of accumulation, like maybe I can try to go up a little bit. So I think Genevieve is referring to this plot here where we see like telomeric like region that tend to have like on the density chart here tend to accumulate some DMCs. I haven't, to be honest, investigated too much about those regions because they tend to be very gene poor and regulatory element poor, but they tend to be, I think, very GC rich. So you could say that maybe they tend to also, by the sheer nature of the underlying sequence, accumulate more of those DMCs. But I think it would be interesting to dig a little bit more about those regions.
Okay, thank you, Matthew. And now I have time for one more question. And comes from Andre. Hi, thank you for the great talk. Do you have information on the parents' historic history of obesity? Is differential methylation something that can be reversed with weight loss, or is it a lifelong alteration?
Yes, that's a great question from Andre. So the parent historic obesity. So we know about the father obesity, of course, level at the time of the sample collection. From the mother side, we tended to avoid any potential confounding factors coming from the mothers. So I want to say that. So here we're looking mostly at the effect of the paternal side of obesity. Regarding the reversing effect, so there's very interesting studies. I think one of the first one was from Romain Barès, where he probed the methylation level in obese individuals that were undergoing bariatric surgery. And he observed that there was indeed a change in the methylation at some loci after the surgery versus before the surgery, so associated with the surgery and the weight loss. So there is studies that suggest that in the case of obesity, you could potentially act on and maybe reverse your sperm methylome to a healthier state if there's such a thing. But those studies needs to be more pushed and more genome-wide, I think, association. But there's evidence that would go down that road.
Thank you, Dr. Schulz. And with that, I would like to thank again thank again Mathieu for a fantastic talk. He will be able to answer the questions in the Q&A box as we move on to the next to our next speaker for today. So it's my pleasure now to introduce Dr. Sarah Kimmins. Professor Kimmins is a renowned expert in epigenetics and reproductive biology. She holds a Tier 1 Canada Research Chair in Epigenetic Inheritance of Disease, and is a professor of pathology and cell biology at the University of Montreal. Dr. Kimmins spent the first 19 years of her academic career at McGill University, where she remains an adjunct professor in the Department of Pharmacology and Therapeutics. Dr. Kimmins earned her PhD from Dalhousie University, where she explored the molecular mechanisms regulating the estrous cycle and pregnancy receptivity. She then completed postdoctoral research under the mentorship of Dr. Paolo Sasson-Corsi using transgenic models to investigate epigenetic mechanisms governing spermatogenesis in fertility. Her current research focuses on the molecular mechanisms of epigenetic inheritance and how paternal environmental influences how paternal environment influences embryo development and disease transmission across generations. In addition to her research, Dr. Kimmins is the co-director of the Developmental and Intergenerational Origins of Child Health Research Network in Quebec, and has also served in leadership roles such as the Andrology Chair for the Canadian Fertility and Andrology Society. She is also a working group member of the International Male Reproductive Health Initiative. funded by the European Society of Human Reproduction and Embryology. Dr. Kimmins' groundbreaking research has earned her numerous international accolades and continues to advance our understanding of epigenetics and its implications for human health. Please join me in a warm welcome for Professor Sarah Kimmins.
Thank you very much for that introduction. And I'm really happy to be here and for the opportunity for Matt and I to present some of the latest research from our team. I'm going to carry on in some ways the theme of obesity in this presentation, and I'll move from our investigations using animal models to the second half of the talk. We'll focus on primarily on our human studies. So lately there has been a shift in the parental burden of preconception health. So mothers have unfairly been the focus when it comes to planning for pregnancy and child health. Women have long been advised.
Sarah, sorry
Yeah.
to jump in. I'm not seeing your slides. If
Really?
Yeah, just
Oh,
if you have
okay.
slides, maybe you're doing a
I do have slides. So that's a problem. Am I allowed, does something need to be pushed on your end to allow me to share my slides?
I'm not sure. Maybe our hosts at Active
No,
Motif.
it should be fine. Do you have multiple monitors?
No.
Yeah, you
Try
should just
again.
have
I've
you should
stopped
just
it.
have shares. You should be able to share your screen.
You see it now?
No.
Oh, okay. Wait. The share screen, I thought that was controlled on your end.
It's in the bottom of
Yeah, I've got it.
There
Sorry
we go.
about that.
Yeah, coming now. I see it now.
Yeah.
It's not in. There we go. Perfect.
So where I left off. So in general, when we think about pregnancy and the health of the next generation, a father's preconception health has long been ignored. But evidence from rodent studies suggests that a father's preconception health, his BMI, his diet, and exposures to toxicants can all impact not only fertility, but also embryo development. And in turn, this can lead to an increased risk for disease in the offspring and perhaps even across generations. So strong evidence exists in animal models that epigenetic inheritance is involved. But what drives epigenetic inheritance in humans remains unresolved, although we likely think there are similar epigenetic mechanisms. And I'm going to show you some of the work that we've done to support that there is a conservation between mice and men in terms of routes to disease transmission in the next generation. So for those of you that don't know about the sperm epigenome, it's very unique and very different than what you see in a somatic cell. So the sperm epigenome that you can see here in this schematic, you have the eviction of the majority of histones, and you have this very unique packaging where you have sperm-specific proteins known as protamines. And then there are still some histones that are retained. And so you have about 1% of histones retained in mice and about 10% of histones retained in men. You also, of course, have DNA methylation, which we just heard about, and small non-coding RNA. So these are all implicated in the transmission of environmental information from the sperm to the next generation. My focus today is going to be on the histone modifications. And so histones we know are retained in sperm at genomic functional regions that include promoters, enhancers, intergenic regions, repeats, and transposable elements. We think of these regions and being involved in regulating gene expression during spermatogenesis. So there's a map for transcription during spermatogenesis. Then there's a map that is also transmitted by the sperm epigenome to persist in the early embryo that's important for placentation and embryogenesis. So in animal models, we know that if there are disruptions to the epigenome at the level of DNA methylation, histone modifications, or small non-coding RNA, these can all impact embryo development and offspring health. So the mark that we're most interested in and have focused the most on in our studies is histone H3 lysine 4 trimethylation. And so this is a gene activating mark, and it's found throughout the sperm epigenome. And we've shown that it's actually critical for fertility and embryo development using a transgenic mouse model where we disrupted the level of enrichment for H3K4 trimethylation in sperm. We've also shown that we can alter the enrichment levels in sperm in mice by changing folate availability. so on a folate deficient diet, and in mice that are obese due to being exposed to a high-fat diet. In a recent study done by a PhD student that graduated from my lab, Ariane Lismer, she showed for the first time that H3K4 trimethylation is transmitted at fertilization to the embryo, where it associates with altered gene expression and offspring development. In one of our first published human studies, we showed that H3K4 trimethylation in sperm from men, when exposed to high levels of the endocrine disrupting toxicant DDT, is altered, and it's specifically altered in genes that are related to embryo development and neurogenesis, for example, much like Matt detailed in his presentation. So how does this occur? So how do these environmental factors interact with the epigenome to change that information there in response to the environment? Well, one way we know is through the one carbon cycle. So the availability of methyl donors alters the level of SAM, which is the universal methyl donor, which in turn can impact DNA methylation and histone methylation. Diet can also affect the availability of acetyl-CoA, and we know that changes in these epigenetic profiles lead to altered gene expression, and we know this from studies of human disease, such as cancer, as well as our animal models. So is there a role for the paternal epigenome and BMI in the epigenetic transmission of disease? So two questions we're going to focus on is extending something that Mathieu suggested in his last slide is, what is the role of H3K4 trimethylation in terms of placenta and embryo development in mice and men? And if you alter the sperm epigenome, can it be repaired? So the placenta, when we think about the placenta and problems with placenta development that lead to things like small for gestational age babies and obstetrical complications like preeclampsia, we usually relate these back to maternal health. But in actuality, placenta development requires correct paternal gene expression. And that was initially shown by some groundbreaking studies that looked at paternally expressed imprinted genes that were done, for example, by Azim Sarani was one of the first studies. So if you look at this diagram over here, in blue, you see the trophoblast, and various types of trophoblast cells are pointed out. When that trophectoderm is forming, that's the first lineage segregation that occurs in embryo development. That leads to that layer of trophoblast, and they form the major endocrine compartment in the placenta. So they're responsible for the production of neurotransmitters, including serotonin, dopamine, norepinephrine, and epinephrine. And because of this, there has been a term coined, the placenta-brain axis. And so it's been shown in animal models that if you have abnormalities in the development of the placenta, this is linked with neurobehavioral and metabolic disorders in the offspring. So there's a tight connection between the paternally transmitted epigenome, paternal gene expression that drives that first lineage segregation, and then a route to disease in the offspring mediated by developmental abnormalities in the placenta that alter that neurotransmitter communication, in turn leading to these disorders. So what do we know about exposures that have led to changes in placenta development? So some of what we know is summarized here. We know that exposure to alcohol, cannabis, and high-fat diets can induce changes in different layers in placenta development. So for example, the labyrinth layer that you see here at the bottom has been shown to be altered in exposure to those factors. And then a low protein diet increases the junctional zone, such as you see here. So you get a change in the ratio of cell types and their differentiation. So this has mostly been tracked back to changes in imprinted genes. But what about other regions of the genome? What about other epigenetic marks? How do they influence placenta development and gene expression? And so we conducted a study in collaboration with the Sloboda lab to investigate that. So to provide some background of that, Anne-Sophie Pepin, who recently graduated from the lab, who is now a postdoc in Germany, she looked at whether our favorite mark is sensitive to being obese. So what she did is she put males on a high-fat diet for a couple spermatogenic cycles. She collected the sperm. She performed chromatin immunoprecipitation, and she looked for changes in enrichment in comparison of the high-fat diet to the low-fat diet. And that's what you're seeing here on these heat maps. And so indeed, we found gains in H3K4 trimethylation enrichment and losses. And then if you look at the GO terms, these were involved in things like development, placenta formation, inflammation, glucose metabolism, and lipid metabolism. All things you would expect to find in terms of what we know was impacted in a physiological response to being obese. So then working with the Sloboda lab and their mouse model, which is slightly different from ours in terms of the diet, they feed a high-fat diet, but then the control is a chow diet. In their model, what they had already shown in terms of the placenta were some interesting physiological changes and gene expression changes. So these are summarized here. They had altered placenta development that was reminiscent of hypoxia because they had changes in placental vasculature. They had altered fetal hepatic gene expression in that model and impaired offspring whole body energy metabolism. So they did a targeted gene expression analysis, and then they sent the entire placenta to us, as well as the sperm. And so we also did chip sequencing on the sperm because we wanted to compare our findings in our model to theirs. And interestingly, there was some conservation between the regions that we found were altered in our model and theirs, suggesting that it's a somewhat robust effect in terms of alterations to the sperm epigenome. And so then Sophie looked at what happened at the level of gene expression in those placentas. And so the results are shown here. So first off, most interestingly, there are sex-specific differences in gene expression, and you can see that on the heat map. So there were regions that were overexpressed or genes that were overexpressed, and also regions that were reduced in gene expression. And if you look down here at the GO terms, they were implicated in things like brain development, neuron morphogenesis, angiogenesis, protein transport, transcriptional regulation, angiogenesis again, neuron projection again. So consistently, there were effects on genes that are involved in neurogenesis and angiogenesis, both in the male and female placentas. Now, when we think about our obesity male models, so we did do a transgenerational study and looked at whether, and that's published, whether there were effects that were transmitted and consistent in the second generation. And so we found that there were no effects in the second generation, and that in the first generation, there were sex-specific effects where the males were much more affected in terms of things like insulin sensitivity than the females. And so part of this change in, or these differences in response to paternal obesity at the level of the sex could be related back to these differences in gene expression that you see in the placenta. On the bottom here, you can see that there was also a lot of deregulation of imprinted genes, those that are both maternally and paternally expressed. And of course, we know that imprinted gene expression is essential for placenta development and function. To investigate whether there were actual changes in the type of cells and the amount of cells in the placenta, and Sophie did a deconvolution analysis. And what she found was that in female placenta, there was a decrease in invasive spongiotrophoblast, and then in both male and female placentas, there was an increase in the female in spongiotrophoblast and an increase in the male. So these included cells being affected that are involved in glucocorticoid metabolism and cells that are essential for vascular remodeling. So again, this fits with the phenotypes that we observe at the level of the placenta and in the offspring. So now let's look at some of our data in men. So the first question we needed to answer as we moved our studies from mice to men was, is our favorite mark, H3K4 trimethylation, similar in terms of where we find it in sperm? So to answer that question, we used a reference population and a pooled sperm sample that was validated with sperm samples from individual men, 7 individual men, and we did a deep sequencing for H3K4 trimethylation. We also did whole genome sequencing for DNA methylation, and interestingly found that where you have intermediate DNA methylation and high levels of DNA methylation, this does overlap with where we find H3K4 trimethylation peaks. So like we find in mice, we find H3K4 trimethylation present at promoters, enhancers, exons, introns, intergenic regions, and also at transposable elements. When we overlap where we find H3K4 trimethylation peaks in sperm with inner cell mass, you can see that there is a strong overlap. And there is also an overlap with gene expression, and we have those findings published in the paper. So overall, it looks like H3K4 trimethylation is similar between mice and men. And now we want to know, is it similarly affected by environment? Can it be implicated in fertility? And can it be implicated in disease transmission in the next generation? So looking a little more deeply at the relationship between H3K4 trimethylation and sperm, the inner cell mass, and placenta, we're looking at scatter plots here where on the y-axis you have where you find H3K4 trimethylation and the inner cell mass at promoters, plotted against where you have H3K trimethylation in sperm, plotted against where you have gene expression in the inner cell mass. And so you can see that 43% of our promoters that are marked in sperm overlap with where you have H3K4 trimethylation in the inner cell mass, and where you have very high levels of gene expression. And similarly, we find the same for H3K4 trimethylation in the placenta. So this suggests that it does have important roles in terms of governing gene expression in the early embryo, as well as in placenta formation. Next, we wanted to know whether it can be altered by the environment, and is it related to fertility, and do any changes there in response to the environment, in this case being overweight, linked to potential disease transmission through changes in embryo development and placenta development. So we recruited men from the Create Fertility Center in Toronto, as well as from the Schulz Fertility Clinic here in Montreal. And we collected clinical data that included BMI, age, semen analysis, lifestyle, things like drug use, including cannabis use, alcohol use, and smoking. We had a look at the fertility history, i.e. did they father a child before, how recently, and we also had the clinical outcomes. And this is things like embryo development, embryo quality, pregnancy, and miscarriage. And so just to keep in mind that we didn't have this information available for every single sample, but where we did, we have included it in our analysis. And then we had a subgroup where we looked at the effect of BMI, being overweight and obese. And on these men, we collected information specific to folate metabolism as well. So we had data on dietary intake, as well as data on RBC, folate, homocysteine, and vitamin B12. So first, let's look at what we find in terms of exposures, things like age, BMI, and poor lifestyle, and whether these actually impact H3K4 trimethylation levels, and whether we can find unique signatures. So first, we wanted to know if we could find a unique signature for infertility. So for this analysis, we compared healthy sperm donors and their sperm H3K4 trimethylation profile to clinic patients that were diagnosed with either idiopathic infertility or athenozoospermia, which is related to motility, or abnormal sperm formation, which is defined as teratozoospermia. And what we found is that there was a common infertile signature in the sperm of infertile men. And this occurred at genes that are involved, of course, in spermatogenesis, including genes involved in sperm function, fertilization, and embryo development. We also identified in infertile men a unique signature associated with being overweight or obese. In men that are of an advanced age, we also found a unique signature suggesting there could be an epigenetic clock in the sperm. We know that this has been found for DNA methylation, in fact. And then we also found that a poor lifestyle, whether it's cannabis use, misuse of alcohol, or smoking, also shows a unique signature in the sperm in infertile men. So what about being overweight and how this may be implicated in disease transmission? And this is particularly important because we know that more than half of the men that come to fertility clinics that are diagnosed with infertility tend to be overweight. So it's a problem not only for their fertility, but also for the health of the next generation, as we saw from our animal models and as is suggested by the work of Mathieu that he showed looking at DNA methylation. So here we plotted H3K4 trimethylation at the inner cell mass promoters against where we have differential enrichment in H3K4 trimethylation at sperm promoters and overweight and obese men. And you can see that there are promoters that bear alterations that associate with high levels of gene expression in the inner cell mass. And then in the scatter plot on the right, you can see similar findings when we plot against placenta promoters and gene expression in the placenta. So this suggests that, like we see in mice, being overweight can alter their fertility and has the potential to influence embryo development and placentation. And something that's important to remind you of in the interpretation of these findings is that when you have an overweight father, you're more likely to have obstetrical complications. And this could be due to influences on the placenta. You're also more likely to have an increased incidence of miscarriage. So looking more deeply here, we did a GO analysis, and you can see that we find genes that are implicated in things like cholesterol metabolism, embryonic development, and trophoblast differentiation. And below, we're showing you examples of some tracks. So on the top, you have the peaks from men with a normal BMI across the tracks, and then you have the peaks from H3K4 trimethylation at these genes in sperm from overweight and obese men, plotted against the inner cell mass, plotted against the placenta. And you can see that in comparison to men with a normal BMI, you have significant gains in enrichment in this mark for the enzyme ACLY that's implicated in lipogenesis and cholesterol genesis. Similarly for MBD3, which is a component of the NERD repressive complex that has a role in embryonic development and pluripotency, you can see there's also gains in H3K4 trimethylation. And then looking at Gen B, you can see also gains in comparison to normal BMI men. So these findings seem to confirm what we find in mice, that there could be similar mechanisms impacting fertility and the next generation that are mediated by H3K4 trimethylation. So the next question is, can the sperm epigenome be repaired by targeted interventions? And so here I'm going to show you some of the work from our mouse model. So this builds on research that had been done by Ariane Lismer. So she fed males a folate deficient diet, and she found that there were developmental abnormalities, and she used skeletal development as a readout. And so we had birth defects that included things like underossification of the skull, malformation of the skull, elongated snouts, for example, and numerous other changes in skeletal malformation. And these were associated with changes in H3K4 trimethylation in the sperm, where there were both gains and losses. And then most importantly, she showed that H3K4 trimethylation that is altered by folate deficiency in sperm is retained at some of those marks in the 8-cell embryo and overlaps with where you have abnormal gene expression in embryos sired by folate deficient males. So building on that research, Romain Lambreau and Christine Lafleur have again used this folate deficient diet, exposing males from weaning throughout their adult life, and then bred them to females on a regular diet. And so males were exposed either to a folate sufficient control diet, a folate deficient diet, or for 10 weeks after the folate deficient diet, then exposed to a folate replete diet. And we call that the rescue diet. And so when phenotyping the E18.5 skeletons, what they found was that you did have an increase in skeletal defects in the folate deficient model, confirming the work done by Arian. And that in the folate deficient rescue, you had a reduction in these defects. And the number of defects for severe skeletal abnormalities was also reduced in the rescue compared to the folate deficient. So could that be occurring through the sperm epigenome and our favorite mark, H3K4 trimethylation? So here on this line plot here, we're looking at regions that we found were differentially enriched. And on this here, we're looking at regions that were decreased in H3K4 trimethylation in response to the folate deficient diet. And you can see that there were 232 regions that were down in the folate deficient diet and compared to the folate sufficient diet. And there were these regions appear to be rescued in the folate rescue diet. And then if we look at a global view here, you can see that we have similar findings showing this. This is a density plot showing changes in the overall enrichments of our mark in the different diets. So next, we're looking here at the regions that gain an enrichment, and you can see that similarly, the regions that gain enrichment in a folate deficient diet appear to be a subset of them corrected in the rescue diet. When we look at the GO terms, we find that the regions that are both up and down in terms of H3K4 trimethylation in those rescue regions involve genes involved in development, reproduction, and neurogenesis. So that provides hope that the sperm epigenome in terms of sperm is not completely set, that there is some dynamism there, that you can change the sperm epigenome, and that potentially in men, like you have in mice, you could change a lifestyle and rescue spermatogenesis. And of course, in men, we know that spermatogenesis takes place over 3 months. So if men are thinking about conceiving, well, then they could adopt a very healthy lifestyle and perhaps undo some of the damage that had been done the years before. So here I'm just showing you a schematic summarizing what we know mostly from animal models in terms of mechanisms of epigenetic inheritance. So we know that you can have these environmental exposures and they change DNA methylation, non-coding RNA, and histone modifications. These are likely leading to changes in TADs, so changing gene regulation. And there's most likely interaction between these 3 epigenetic horsemen. So DNA methylation and H3K4 trimethylation, as I showed you, can co-occur in certain regions in the sperm epigenome. And we know that the expression of non-coding RNA in particular regions can be controlled by the levels of H3K4 trimethylation. So some of these changes can lead to changes in transcription factor binding, and we know from the work of Corses that transcription factors are retained on the sperm epigenome and transmitted at fertilization. So there's still a lot of work to do. As you can see from what I've presented, we're really reliant right now on association, and so we need to get at mechanism. And so the development of epigenome edited models that we've started to do in our lab and is being done in other labs will really help us get into what are the exact mechanisms that are involved in this transmission and that lead to disease. So there's a lot of opportunity here to improve the health outcomes for children by focusing on men's health and the link to the sperm epigenome. So we really need to launch public health campaigns that target youth, men, and couples, with messaging that dad's health matters too when it comes to having healthy children and to optimizing fertility. So I'd like to end by thanking my team, and highlighted here you can see some of the key people on my team that contributed to the work that I showed, as well as our key collaborators and our funding and recent alumni. And I'm happy to take some questions.
Wonderful. Thank you, Sarah, for an excellent talk and very compelling data and message that you're communicating. Again, just as a reminder, please ask your questions in the Q&A box. And to get things kicked off, I'm going to ask a technical question. So I noticed in your I think in the second part of your talk, that you had observed co-association of H3K4 trimethylation and DNA hypermethylation.
Yeah,
And I guess that is I guess that is unexpected given the mutual
it is.
exclusivity
Yeah.
of K4 tri and DMTs. And so does that tell us something about the process of deposition of K4 tri, or can you talk about how histone mods are actually mechanistically regulated in the sperm?
So just like in a somatic cell, you have the same deposition of these marks by the same machinery, the same epigenetic machinery. There are some testes-specific versions of histone variants. And so there could be different proteins that interact with those histone variants to establish the sperm epigenome that have yet to be identified. But the sperm epigenome, as I mentioned, is unlike somatic cells. So you're right in that where you have H3K4 trimethylation, usually that repels the DNA methyl transferases that deposit DNA methylation. So why they co-occur in sperm is actually a mystery.
So thank you, Sarah. I see Jennifer has her hand up as a panelist. Please, Jennifer.
Hi, yeah, Sarah, it was really fantastic work. I was curious to know, do any of the like diet interventions or treatments that you mentioned, are they so severe as to actually cause like a reduction in the number of offspring that are produced? Like, do they actually cause like placental and mid-gestation placental insufficiency, or are they not that severe?
They're not that severe.
Okay.
So
Yeah.
the males are in general pretty healthy. We didn't see a change in things like sperm count. And it's really hard to disrupt fertility in male mice. So we've done various exposures that we really don't see that. We do see it in our really severe genetic model, where we've altered H3K4 di and trimethylation, but not in these exposures to the diets.
Okay. Thank you.
Yeah, we did have actually early on when we did a lifetime exposure to folate deficiency, so beginning in utero, we did have changes in implantation rates and pregnancy loss, but it's less severe in effect when we do our most recent diets, which we're starting at weaning.
Thanks.
Wonderful, thank you. So I'm going to move on to some questions that are coming into the Q&A. And just as a reminder, we probably won't have time to get through all of them. I see that we're at the top of the hour. And so I'll ask a question here from Isabel. Great work, Sarah. Many thanks. I'd like to ask if you could elaborate on the possible role of somatic cells, which mediate metabolic nourishing in the testes, for the epigenetic effects and their rescue. Also, could you comment on what protamines in the sperm contribute? Thank you.
Okay, so I'll start with the Sertoli cells and the somatic cells. There really hasn't been anything done in terms of interaction there and how that can be altering the germ cell epigenome. And that's a great idea to do it. And as you're probably aware, there is quite an accumulation of work showing that there is somatic germ cell interaction in the epididymis, which is essential for some modes of epigenetic inheritance. So yeah, this is an uninvestigated area where we might find that there are similar things happening in terms of things like vesicles depositing things into or on the sperm. And you do have these bridges of communication in spermatogenesis.
Wonderful. Thank you, Sarah. So I think we have time for one more question. This question comes from Julie.
Oh, the protamines. I didn't get to the protamines. What are they doing? So Sue Hamoud is doing some really nice work on the protamines. And I can't remember right now the details, so I suggest to look at her papers, but she's done some really nice recent work on that.
Great. Thanks, Sarah, for that clarification. So last question from Julie. Thanks for your presentation. This is a fascinating and complex topic. Did you try or do you plan to detect sperm DNA methylation and placenta DNA methylation in your folate rescue models?
No.
Both?
Those models are, we actually, what we have coming next is Romain and Christine have used genetic crosses, so we can look at specifically the maternal and the paternal genome contributions there. And that required so much hard work and so many mice that we are going to stop there. We'll probably do some chromatin in the embryo. We've done the gene expression with the results to come, but no, I don't think we'll be getting into DNA methylation. So hopefully somebody will. I know Jaquetta Trasler has looked at it and published nice work there on folate supplementation of the father.
Wonderful. Thank you, Sarah. And with that, given in the interest of time, I think we'll end it here. And I would just like once again to thank both speakers today. It was very fascinating and fascinating talks looking at the implications of DNA methylation in the paternal sperm. So thank you again, and thank you all for joining. And with that, we'll end it.
Okay, great. Thanks so much.